In this lecture, we're going to be talking or taking a tour of an animal cell via the process of protein production. Um, and so sort of before we get started with that, this is an animal cell. And let me just ask you, do you think that this is a prokaryote or do you think that this is a eukaryote? And how would you be able to tell the difference? Let's say, for instance, you don't know it's an animal cell. It's just a cell that you happen to be looking at under a microscope. Is it prokaryotic or is it eukaryotic? Well, it's eukaryotic, and the way you know that, aside from the fact that it's an animal cell, the way that you know that is because inside of this animal cell are all of these organelles. There's all of this stuff in here, some of which you can see in the microscope. The main one that you'll be able to see in the microscopes, as we'll see in lab, is the nucleus. You might be able to see some other organelles, but the main one that you'll be able to definitely resolve is going to be that nucleus. You'll also notice that there's a bunch of organelles in here that you're not going to see talked about very much in this video. This is, again, just a tour of protein production, and you saw in the last lecture series about protein and why proteins are so important. And so we're going to talk about building those proteins today. But if you see things on here that you don't necessarily hear me talking about, that's okay. We're going to talk about them later on. So again, here is your nucleus. Inside of the nucleus, you have this thing called the nucleolus. Okay, the nucleolus is involved in the production and assembly of ribosomes. Inside the nucleus, you also have these strands of uncoiled DNA. So you have all these strands of DNA, and again, DNA lives inside the nucleus of a eukaryote. And you also have these nuclear pores, and the nuclear pores are going to allow for the exit of a molecule called RNA, which we're going to talk about shortly here. So again, we are talking about the production of protein, and the protein production begins within the nucleus of a eukaryote. It begins within the nucleus of a cell. So that nucleus is, again, it is a membrane-bound organelle. It is a membrane-bound organelle, and inside of there is DNA. And DNA is the recipe book for proteins. So DNA is the recipe book for proteins. And what happens is the cell needs proteins, or maybe the organism that contains a cell needs proteins. And so that cell, or maybe that organism, will go to the nucleus and say, hey, you know, I need a protein that does such and such. I need an enzyme that can help me catalyze this reaction, or whatever it happens to be, whatever that body, whatever that cell happens to need, whatever that organism happens to need. It goes into the nucleus and it makes a copy of the recipe for the protein that it needs. That protein, or that, I'm sorry, that recipe is copied in the form of RNA. So again, just to orient you to where we are, we're inside the nucleus. We have all of these recipes written down in the form of DNA. When a piece of protein, or when a protein is needed, the cell will make a copy of the recipe for that protein. And that copy is encoded in what's called RNA. Specifically, it's encoded in something called mRNA, or messenger RNA. So we're inside the nucleus. We have all of these recipes for protein in the form of DNA, and we need to make a copy of them in the form of mRNA, which can then leave the nucleus. Okay, remember I was talking about those nuclear pores. Well, the mRNA can actually exit the nucleus via those nuclear pores and can go into what's called the cytoplasm. The cytoplasm is just everything inside of the cell. Okay, that is the cytoplasm. So here's our whole cell that's sort of been cut in half, and then they take up this nucleus and they zoom in on it for you. So here you have the nucleolus, which again is involved in the production of ribosomes. And then inside of the nucleus, not the nucleolus, but inside of the nucleus, you see you have these little strands of DNA. They look like little, I don't know, spaghetti strands in here. And when you want to make some kind of protein, you're going to copy that DNA into mRNA. And that looks like this. Little piece of mRNA. And that mRNA is actually able to exit the nucleus via the nuclear pores. Okay, so RNA is able to exit the nucleus via nuclear pores. And this is just an awesome picture of a nucleus, and you can see the nucleolus is this extra dark spot inside the nucleus here. So after you make your mRNA recipe, or you copy down your mRNA recipe. So just to make sure we're all on the same page here, the nucleus contains all of these recipes for protein. And then what your cell does in this process called transcription is they write down a copy of it. Because you don't want to take the original copy with you outside the nucleus. It can be damaged out there. It can get messed up out there. So you just write down a copy of it in the form of mRNA. That mRNA is actually able to exit the nucleus, and it's going to go to endoplasmic reticulum, which is another organelle within your cell. Okay, so the endoplasmic reticulum has two regions. There is the smooth endoplasmic reticulum, and there is the rough endoplasmic reticulum. The rough endoplasmic reticulum is called the rough endoplasmic reticulum because it has all of these ribosomes sort of studded all over it. Okay, and this is the location where much of the protein that your cell creates is made, is on the rough endoplasmic reticulum. Okay, so protein is made by the ribosomes. Okay, and the ribosomes are on the endoplasmic reticulum a lot of the time. Ribosomes don't necessarily need to be attached to an endoplasmic reticulum. They can also just be free within the cell. Okay, and like I said earlier, ribosomes are made in the nucleolus. The reason that you might want to make a protein on the endoplasmic reticulum is because it is processed and folded inside the endoplasmic reticulum, and it can also go to other places. So let's say you have a protein that's needed somewhere outside of the cell. Let's say you have a protein that's needed, maybe on the outer membrane of the cell, or maybe it's needed, for a cell nearby. You put it inside the endoplasmic reticulum, and the endoplasmic reticulum is actually going to help that protein to be able to leave the cell itself. So here's kind of a picture of how that process works. So here again is the entirety of the cell. There's your nucleus here. There's your nucleolus sort of depicted in the middle. And outside of that, you wind up with the endoplasmic reticulum. Okay, here is your endoplasmic reticulum. It's sort of right outside the nucleus to make this process of getting RNA to it easier. Here's a micrograph that's been blown up depicting the endoplasmic reticulum. And I'm actually going to zoom in on this lower part down here to make it a little bit easier to look at. So after your RNA leaves the nucleus, it's going to dock onto a ribosome. Okay, and it's going to start creating this amino acid chain. Remember from the previous section, a chain of amino acids is also called a protein. Okay, a chain of amino acids is a protein. So this ribosome is going to start excreting, well, it's going to start creating this amino acid chain, and we're going to talk about the details of how all these things work later on, but that's for a later section. So we're going to talk about the details of how you construct this mRNA. We're going to talk about the details of how the ribosome puts together this amino acid chain, but that's for later. For now, it's sufficient to just know inside the nucleus, you're making a copy of that recipe. That recipe is leaving the nucleus via the nuclear pore. It's going to find this thing here called a ribosome, and then it's going to start creating this amino acid chain. When the ribosome is done making that amino acid, it's going to kind of come take it and drop it off inside of the endoplasmic reticulum. which is kind of like, well, why are you doing this? You just made this protein that you might need in the cell. Why are you putting it into this other organelle? Well, this is done for a few reasons, and it's because the endoplasmic reticulum is, for one thing, it's going to help a lot of proteins fold up. It's going to add sugar side chains to those proteins. It's also going to be serving some quality control functions. If your protein is completely messed up, the endoplasmic reticulum can help to fix it, or it can actually destroy it so you don't wind up with a messed up protein running around inside your cell or even outside your cell. And one of the more important parts of the endoplasmic reticulum is that it puts these proteins into these vesicles so that they can be moved elsewhere, so that they can go to this other organelle called the Golgi complex or the Golgi apparatus, which is going to do some other stuff to it. So in the cytoplasm, transport vesicles, so just to remind you, this is a vesicle, okay? So the ribosome created this polypeptide, this protein, dropped it off inside of the ribosome, inside of the endoplasmic reticulum, excuse me. So the ribosome created this protein, dropped it off inside of the endoplasmic reticulum. The endoplasmic reticulum helped to make sure it was all nice and folded up, maybe added some sugars to it, and then put it inside of this thing called a vesicle. So this is actually going to bud off right here. This piece of the endoplasmic reticulum is going to bud off, and then this is going to be taken somewhere else. This protein is going elsewhere. So that vesicle is going to then go move through the cell and fuse with this thing called the Golgi complex, which modifies, sorts, and ships these proteins to their final destination. So here's a picture of the Golgi complex. Here it is here. So that transport vesicle that budded off from the endoplasmic reticulum with this protein inside of it comes in and fuses with the Golgi complex. So you can see this protein was never out inside of the cell or inside of the cytoplasm. It was never free in there. It was always in this vesicle. It's going to come and fuse with this Golgi complex, and this Golgi complex is going to continue processing it, potentially modify some of its side chains. It's going to change the protein up a little bit, and then it's going to take and send that protein to wherever that protein needs to go, whether that needs to be the plasma membrane, whether that needs to be inside of the cell itself, and then maybe the protein just needed some modification, or it's going to be external. It's going to be outside of the cell. So once that protein is finished being created, so you start in the nucleus, you have all these protein recipes, you write one down in the form of mRNA, you send that to the ribosome takes that to the endoplasmic reticulum, the endoplasmic reticulum does quality control steps, it maybe adds sugars to that protein, then it buds off from the endoplasmic reticulum where it is taken then to the Golgi complex. The Golgi complex further modifies it and puts shipping labels on it, basically tells it where it needs to go, and then it is sent off from the Golgi complex via this process of exocytosis. So remember earlier, we had this vesicle thing at the end of the endoplasmic reticulum here. This was going to go to the Golgi, right? So then you get to the Golgi, and this vesicle fuses with the Golgi, and the Golgi is saying, okay, protein, you need to go and exit the cell. You need to go somewhere. Well, that process is done via exocytosis. And that's when the vesicles are actually fusing with the outside of the cell and being released. So I should have done that whole review on this slide. Sorry about that. Instructions are copied into mRNA. mRNA goes to the ribosome goes to the endoplasmic reticulum, which does more stuff to the protein as it's coming out of the ribosome. Okay. Then it is put into this vesicle, which is taken to this Golgi complex, and the protein is dumped off inside the Golgi complex. The Golgi complex further modifies and puts these proteins into yet another vesicle and sends them on their way. So when this is the outside of the cell, by the way. So this is the plasma membrane of the cell. This is all stuff in here is all happening inside. When this vesicle fuses with the plasma membrane, when this vesicle fuses with the outside of the cell, that is a process called exocytosis. Okay? Exo like exit. Exocytosis is the process by which proteins are going to be moving from the inside to the outside of the cell. So in the next lecture, we're going to be talking about some of the other cell structures within a eukaryotic cell. We're going to talk about some of the other organelles that are in there that we didn't get to this time.